welcome everybody to the second day of the TropiBEF workshop 2020. It's a pleasure to be here, and today we have the satisfaction of having as the opening talk of this workshop, Dr. Gislene Ganade. Therefore, I would like to thank her for having accepted our invitation to give this talk for our workshop about the BrazilDry project. Dr. Gislene Ganade obtained her PhD in ecology from the Imperial College of London in 1996, in which she was investigating seedling establishment in the Amazon forest. Currently, Gislene Ganade is a professor at the Ecology Department of the Federal University of Rio Grande do Norte. She has worked with restoration ecology since 1992, and her research aims at developing new technologies applicable to the restoration of degraded tropical and subtropical ecosystems. Well, I have the need to publicly express my admiration and respect for Dr. Gislene Ganade, who was my PhD supervisor. She's an inspiration for me because of the high quality science she conducts, and also because of the kindness and generosity she has with people who work with her. So welcome, Dr. Gislene Ganade, the stage is yours.
Thank you very much, Leo. Thanks for a very warm welcome. I was almost crying here, so thank you. So I will share with you some of the challenges and solutions we found in our dry tropical forest experiment to help people think in this big net of experiments in tropical areas that we could conduct in Brazil. So the BrazilDry experiment actually was the first experiment which entered the 3D net, which is a net of experiments on diversity and functioning that are spread all over the world. And this was made in Caatinga seasonally tropical dry forest, which is a very squeezed environment. We know that the dry surfaces in the earth are very large, and Caatinga is here. We have this northeast Brazil, a very large area, 800,000 kilometers of semi-arid forests. And this area is very sensitive to global climatic variation, so sensitive to climate changes, and some recent works have shown that it can be an important carbon sink too. Also, there's a wide diversity and a very unique diversity for this forest. All these forests all over the world. Caatinga has its unique sets of plants and also animals that are adapted to the semi-arid regions. What I'm going to tell you a little bit in the beginning is my story, how I came to work with these experiments in Caatinga. In Imperial College in Silwood Park, when I was doing my PhD, was a time when the ecotron experiment was launched. And in '94, there was this paper in Nature that caused many discussions and was very inspiring for a group of, enormous group of researchers that implemented experiments in the field. And by that time, I was thinking, I was doing restoration in the Amazon for my PhD, and I was thinking, oh, what a cool idea if we could work with diverse stability and restoration in high diverse environments. And I did many postdocs during this time in Australia, in Brazil, in Rio, many places. And then I got my first position in a university, and I thought, oh, this would be a neat idea to have this type of, there in South Brazil, we have this Araucaria subtropical rainforest. So I was trying to understand that system and to build these biodiverse functioning experiments in this type of forest. And then when I had everything set, I contacted Andy Hector, who was he was a student of the same supervisor. So we met in Imperial College. He was by that time already organizing with many people this 3D net. And I was lucky to be able to visit the Sabah experiment in Malaysia. There was a group of people from the Chinese experiment too, in the China experiment, the BEF experiment in China. And it was amazing because there they have like 70-meter high trees, and the herbivores are elephants. So if you can do something like that, you can do in any tropical forest. And then by that time when I returned, we just had an offer of a job offer. My husband is a researcher too. And we applied for this job offer in a place we always thought to work. And we actually changed our jobs as professors too in this university, UFRN, which I am here till now, in Natal, and Northeast Brazil, where we have this Caatinga semi-arid forest. And then I had to start, I have to start all over again and try to understand this system and know how to work mainly with restoration, which is a challenge in drylands because you have drought, you have soils and fire and heavily use of land, and you have lots of mortality in these systems. So how is Caatinga? Caatinga is a semi-arid tropical forest, but it has clear dry and wet seasons, and there are long periods of drought, but water is a limiting resource, a very important resource, but soils are reasonably rich. That's why plants can afford to lose their leaves. So plants can lose their leaves in the dry season, and there are lots of cool adaptations like trunks that can do photosynthesis. Those are two species I used in this BEF experiment at the BrazilDry. And also another species I used in the experiment, it has some trichomes and hydrophilic polysaccharides that can absorb water through the leaves. So many adaptations. I have a master's student who's working, she's working now with lentil cells to see if they can use vapor, absorb vapor. Actually, we have some interesting results with the Fabaceae, where there's a trade-off between size and number of lentil cells, so some selection is taking place in this. And also survival of young transplants in restoration is positively correlated with the total area of lentil cells, which would indicate that the lentil cells are important to absorb water in the system. So from all these very cool adaptations, we have this, the roots. The roots absorb lots of nutrients and water too, so the plants can afford to resprout when the rain comes after the dry season. But this tropical forest has been destroyed, and what we have now are 50% of the tropical forest remaining, but in large fragments. This is an article published by colleagues from my university, and these large fragments are suffering continuous anthropogenic disturbance. So this is a PhD student, Dr. Felipe Marinho, finished his PhD, and he has shown that when the area is clear cut, so wood is removed, and with increase in grazing intensity, there is a clear decrease of vegetation cover, which is very important, and it leads to this chronic disturbance, which is another paper published recently showing that in Caatinga you have chronic disturbances in all the fragments that are left. What are the consequences? The consequences are desertification, which is one of the things that just was interesting to think about that, because you have to find some special questions for your biodiversity and ecosystem function experiment, because there are many experiments looking at diversity, but each experiment has their own idea or their own approach that could take the ideas a little bit further. So desertification was something that I was, wow, this is cool, because I don't know about papers discussing, BEF papers discussing desertification. So these areas, the desertification in these areas is spreading. Also, it's a recent work also showing that. This is from 2000 to 2005, desertified areas, and now they are more spread, and we have 16% of the area desertified. And when you think about programs of restoration, here we come with a very large difficulty, because mortality for transplants is very high. 70% is with conventional methods. An example is this project done in Caatinga with enterprise that went there and did everything they could do. It was like $70,000 per hectare, the restoration of this area. And after 5 years, they had 95% of mortality, because this is what you face in situations of heavily drought. So the first thing we had to face was how to increase the survival of our transplants to implement our experiment. So we knew the standard method production would not work properly, and then we used then a method increasing root area for our trees. So this is what Johannes was talking about, the tubes. So we were producing our plants in large tubes, in 1 meter tubes with water accumulating here. The roots would come to the water and plants could grow larger. And after 2 months, we had all these roots using the tubes. And some results of a PhD student also from the lab, Cynthia Pinheiro, and she was showing from the first 3 months that even though the above ground area is short, the above ground part of the plant is short, roots can be really large in many of the species. And also these roots can have lots of stock of amide and sugar, which is very important for the plants to be able to resprout when it faces drought. It loses the leaves and waits until it can resprout. So just showing how we were planting there, we had this plants put in this, small plants put in these tubes. And then we could take out bands around the tubes. It had 2 faces, and make a 1 meter hole deep, put water below so the water doesn't get contact with the soil and loses to the atmosphere. And then we can put the soil again back. And after that, we can remove this part of the tubes and use it again because it doesn't break, and the roots are still remain in contact with the soil. So to test the importance of this technique, we used lots of combinations of treatments that were applied in this national forest in the area we were working. It's an area that has high salinity, so no trees were really establishing naturally for 15 years. And in the wet season, we used 6 species initially and tested many treatments against our large root, irrigation treatment. We had to get a tank there. The agroforest treatment with beans and maize and maize and pumpkin, and the trees here you can see in the middle. And also the application of litter, which can bring lots of spores from mycorrhizal plants. We used all these plant shapes for large and small above ground, large and small below ground. And in the end, we had negligible performance of seeds and doing nothing. So you have to do something to plant there. And seeds are not very good for these areas with high salinity. But we found that our large plants with large roots were no different from the plants that had the standard way of planting for wet forests, even though they were irrigated. So you don't need irrigation for these plants. So when we look at the control plots, what it had in 3 years was a survival of around 70% for plants with large roots and 30% of plants with small roots. And this was our beginning to think maybe this is what we can do to be able to implement the experiment, the BEF experiment in the area. And also when we look at cost and benefit, because irrigation is expensive and water is very important for people to use in the system, the lower cost by benefit was the no irrigation with large roots. So that's what, so it was actually got a Dryland champion certificate for this technique. And then we went to apply this technique in our plant community. In another question that was very specific from this work that was not being discussed much in BEF experiments was the question of facilitation. So we had this question of how plant communities with high diversity could improve performance and then improve the capacity of the community to resist and to desertification, but also how facilitation as a process could increase the performance of these communities too. So we did some tests of facilitation, putting seedlings and seeds below trees and far away from trees in an experimental manner. The first experiment that was done in Caatinga about facilitation was made by Gustavo, who was a master's student in my lab by that time. Now his Dr. Gustavo Paterno is organizing also the seminars like Leo. And this experiment was very important to show that facilitation is a very strong force for our system. And then after that, we then studied many trees that we would use in our BEF experiment for facilitation in Doutora Marina Fagundes' PhD thesis. So she tested 20 trees, and from these 20, we used 16 trees. So with some plants below these trees, target species. So we had all these 20 nurses with plants below and far away from nurses, and we could calculate this relative interaction index to show facilitation or competition was taking place. What is interesting is that there is this gradient of facilitation and competition taking place in this area, so we could use this gradient in our design. So we were testing the relationship between biodiversity and ecosystem function, but using two different mechanisms, trying to tear apart the complementarity mechanism from functional complementarity, which is when species have differential use of resources in space and time, and they have more complex exploitation of resources, and then it causes an overall improvement on ecosystem functioning. And then facilitation, when you have a nurse species there that increases resources as a whole by a hydraulic lift, nitrogen fixation, by improving conditions and microclimatic conditions. And this higher resource availability would improve performance for all species, and it will take to an overall improvement of ecosystem functioning. So the idea is like in this scheme, is that when you have high complementarity and high facilitation, you would have higher biomass going on, and which would be this model here that we were explaining, or possible models that could happen in relation to facilitation and complementarity. So after I had everything done and I had all my plants grown in the greenhouse, I was lucky to be able to be part of the Leipzig meeting of the 3D net in 2014. Michael, Sharon, Lawrence was very kind to bring us to visit the biotree experiments, and everybody was very nice. It was the time I was discussing many details of the experiment, and many important things were said. And I'm listing here because for all the people that intend to implement experiments in Brazil, you have to have random assemblage. I used a random partition design. So you have to have random assemblages. In this case, I did without replacements. So it would account for diversity, species identity, and species composition, and how all these things are influencing ecosystem functions. In this design, all species occur in the same number, which is very important because it's just full randomized. Sometimes you have one species that occurs only once in one plot, and then you don't have the possibility to study the effects of this species fully. All random assemblages are replicated, all the combinations have to be replicated, and the species diversity treatments. The assemblages in our case represented a gradient of community facilitation. And control plots with no planting was something that people said, oh, it's good that you have that, so you have some comparison without planting. And the importance of monocultures, and I'm stressing that because lots of people that are doing restoration and have enterprise involved, they are not very easy with this fact that you can have a many plots with only one species planted there, but it's very important to understand what the species does alone and together with other species, so you understand the mechanisms that are behind this diversity functioning relationship. So the BrazilDry experiment was then implemented. Now we are happy that it received from a DC and PK a project of long duration, which we call a small grant that helps to keep the projects for long duration. It's 4 years, renewable for 4 years, and can go on like that if it's working well. And the experiment design is like, we have these 3 by 3 hectare areas with all this treatment, 1 species, 2, 4, 8, and 16 species. We put the controls too. And one thing was the way we randomized in the plots made that one species had many other species around them. So you could do it as much as possible so you can have quicker results in relation to diversity. So we did the planting in 2016 after a sequence of very dry years. We had an outstanding survival of 73% with these long roots. We were expecting 30%. Plants differ in the way they survived, but some plants were very key, like this Mimosa tenuiflora. This species, Leo, has an important work that shows that this species increases phosphorus retention in soil, nitrogen, and has a multifunctional influence in the whole community. So with this long root technique, what I can tell you that it may be central for planting many types of systems, not just dry systems. We planted with no rain after our planting. Plants went through a very long dry season. They went through an 8 months period of no water, and they had enough resource to sprout. And we had 75% survival when we expected 30%. We have been monitoring the area from 2016 when we put on the floor. The experiment launched to 2020, and we had some flowers going on and some fruits being producing, massive flower production because the species flower very early. And if they are large enough and they have resources that come from the greenhouse development, they can do that in the field in a few years. The measurements we took were survival, growth, and biomass until 2019, but 2020 just survival because of the COVID virus. We measured insect diversity, herbivory, herbaceous cover, and leaf litter decomposition. And from 2021, because we got the Spelling funding, some other funding from Norte and TropiBEF, we are going to measure to survival, litter decomposition, herbaceous species, but also insects, amphibians, reptile, and bird diversity in our sites. Plant ecophysiological parameters too, together with Rafael Oliveira from Unicamp and Lucy Rowland from Exeter University. So just to show some initial results on biomass production, which was the number of leaves produced multiplied by the weight of each leaf, we got the biomass. Biomass production was not responsive for the first 2 years in relation to biodiversity per se, but we had many groups of species that were different from each other. Nevertheless, we had biomass production being significantly influenced and positively influenced by community facilitation. We measured community weighted mean for the plots, and facilitation was important to predict biomass production in these areas. We also measured the net biodiversity effect, which is something that you need the monocultures to measure to see how plants differ in the way they behave when they are alone in monocultures or when they are in mixtures. And we have complementarity effect and selection effect affecting all the groups of species we used. But also an interesting thing when we look at the fauna, all these animals were visiting our sites, but we studied the insects. So insect abundance and richness. And one interesting thing is that we had a significant influence on plant diversity on insect richness, and it was significant. So plant richness, diversity is important to predict other trophic levels richness too. It does, but it doesn't influence survival or herbivore level on the plants you are planting. So you just leave the insects there growing and producing more biodiversity. You don't have to control them with insecticide and so forth. One thing that it was a problem for us was herbaceous layer that was growing there. So when you put it, this is another tip, when you put an experiment in the field, you should visit and measure things in the beginning, like growth, survival, insects, and if you have vines, if you have competition around, measure these things because it helps a lot your plants. So I've been cleaning around the plants from these vines, and I show you that this herbaceous layer is very well adapted to the region. Some plants are really beautiful, but some plants are very nasty with the trees you put there. So this experiment from Jani, who is a PhD student, she removed the herbaceous layer, left it there, and she could see that plants that didn't have the herbaceous layer around actually could grow better in terms of height and leaves, and also leaf area was higher where they didn't have the competition by the herbaceous vegetation. So what we found, the facilitation improves biomass production, plant community restored with high diversity have higher diversity of other trophic levels, such as insects. This higher insect abundance and richness did not increase herbivory or mortality. And also that herbaceous species came to your paradisial plants and you have to clean them in the beginning. We are working with broader views of Caatinga too, but those are very important to, we can see that the diversity mechanism that we see in experiments, local experiments, can have an influence. These mechanisms can have an influence for the whole system. We found mapping by ecosystem services that we have a positive relationship, a significant relationship between biodiversity and ecosystem service for the whole area of Caatinga, and also vegetation stability, which is the capacity of vegetation to turn green, is also related to phylogenetic diversity. So as phylogenetic diversity increase for the whole Caatinga area, vegetation stability increases too. So we are now going to this decade of ecosystem restoration. So this is a perfect moment to launch a net of experiments in Brazil that could do restoration and be implemented with the mentality of the biodiversity functioning experiments. So we could do that in many of our biomes. Actually, we are not having much financial support from this government, but there is a program that encourages this forest restoration, and they have people from different biomes in Brazil that will produce this portfolio of how to restore forests, Atlantic forest, Amazon forest, Caatinga, and so forth. So this could be a step to make the link, the enterprise that have to plant with actual restoration in the field. But it's very important that the scientists are together with these enterprises, because otherwise the enterprises are not going to produce a proper biodiversity functioning experiment. Just to thank all people in the lab, in the scouts, and people in the workers in the national forest that really helped. You never do these things alone, so it was a very nice group of people working together. And I would like to show you, giving a last tip here, which is you should, since the beginning, since the beginning, you should use drones to see how your experiment is going on. And I will show you some small 3 minutes video that shows drone images that are very nice to see how things are developing. So you get, the enterprise are never going to make the plots perfectly. So you have them to build all the plots perfectly and measure and take pictures of your drones. Can you hear the sound?
Yes, we can.
So that's it. Thank you for listening to the talk, and I'm open to the questions. Thank you.